Okay.
All right.
Hi, good evening, everyone. Thanks for coming. So today we'll have Phil. So Phil was born in New Jersey, in Holland Township. So he said he's basically been here all of his life, too long. And he went to Rutgers for undergrad, doing the professional physics option, which is basically the option you take if you plan to go into physics research. And his current research is in biophysics and working on imaging systems for single molecular detection in 3D live cell imaging. And his main interests outside of academia are music and skateboarding. And he's been playing guitar for 14 years. Wow, and have composed and recorded a lot of music. Yeah, I look forward to hearing about it. And skateboarding gives him a reason to go outside. I guess now. That's a good reason. Yeah, yeah, maybe I should be before. Anyway, okay, thanks, Phil, and please take it away. Thanks.
All right, I'm ready to go.
Yep.
Okay. So today, if this notification stops popping up, I might have to do just, can you see that little notification in the bottom right? All right, whatever. We'll just ignore it. So today I will be talking actually mostly about optical tweezers and then surface enhanced Raman scattering. And this is all a part of the quest to image live cells at the single molecule level. After I get through these slides, I can talk more about how we plan to do that, but I wanted to talk about these two basic things here today. So Arthur Ashkin.
Can I ask a question? So that means still now people cannot image single cell? Is that what you mean, or people already can do it?
So we can image cells at the single molecule level using specific techniques having to do with labeling. So like if you wanted to image a very specific protein, you'd get a biochemist to attach a fluorescent label to that. And then you can use some methods which make it blink. And you can use processes like post-processing to isolate those dots beyond the diffraction limit of light. So it's a far field imaging technique that basically cheats to get better precision than the diffraction limit. So people have been doing that for a while, and that is called super resolution imaging, which you can Google. And it's pretty awesome. What people, especially Sang-Hyuk Lee, want to do is full 3D real-time live cell imaging. So in the method I described before, generally what you would do is you would fix the cell in formaldehyde or something, and then you have to do this process to label the specific protein that you want to look at, and then over a period of hours or even days, you image this thing to get these blinking videos that you can then post-process and then make a super resolution image. What we would like to do is do all of that in real time without having to label a molecule specifically.
Wow.
So this is what I'm going to be talking about is one small portion of achieving that. And there are many other things beyond what I have here that are required for achieving that. But this is one of the basic tools for hopefully achieving that. So the first tool is optical tweezers that was invented by Arthur Ashkin. By the way, can you see my mouse?
Yeah.
Okay, good. That'll be my pointer. So he's known as the father of optical tweezers because he created them, and he actually just received the Nobel Prize. And he was the oldest at the time when he received it to receive a Nobel Prize, but then like a year after, someone who was 97 got it. And he actually just passed away recently, so rest in peace. So he did this work in the 70s, and these are images from his paper. and he was able to observe radiation pressure and radial trapping. And what I mean by radial trapping is a force due to light due to light's momentum on glass beads in water that pull the bead towards the focus of the laser. So maybe I should unpack that a little bit. We have a laser going through a lens here. It's focused. Here's the focal point. And then this rectangle is the sample chamber that's filled with water and filled with beads. And what happens is when the bead gets close enough to the laser, it actually gets sucked in towards the focus of the laser. So this was his first observation. And there's two parts to this force that I'll go over more in a minute. There's the radiation pressure force, which pushes it in the direction of the laser. And then there's the gradient force, which pulls it towards the region of highest intensity. So what he thought to do to counteract this radiation pressure force, which would push it down the direction of the beam, is to just take two laser beams and point them at each other. And he was able to observe a stable trap using this method. So there are three cases for analyzing how this occurs. There's the geometric optics regime, which I think is the best first step for understanding this. There's the me optics regime, which I have no intuition for, but it is required for particles that have the same size as the wavelength. And then there's the Rayleigh regime, where the particle is much smaller than the wavelength. So the whole particle kind of acts as if it was in a static electric field. And yeah, R is just the radius of the particle. It could just be the size of the particle, since these are just kind of inequalities.
Can I ask a question?
Yes.
So for previous slide,
can I? Hold on. All right.
And this one. So for what's the minimum size of the say cell or some particle you can image?
Minimum size of a particle for what?
Like for this process.
So let's see. I mean, if you have like a laser, say wavelength 1,000 nanometers, so it's like a micrometer.
Yeah.
And you want to trap a bead, depending on the size of the bead, you can enter any of these regimes. So if you have like a 5 micrometer bead, then you're kind of in the geometric optics regime. Now, these aren't like clear-cut distinctions. Obviously, they continuously vary between each other. You know, as you get closer and closer to each regime, they kind of smoothly move through each other. But if you have a 5 micrometer bead with an infrared laser, you're generally in the geometric optics regime. If you have a 1 micrometer bead with 1 micrometer wavelength laser, then you're in the me optics regime. And then across the volume of the particle, you get these interference effects, which change the forces on the particle. You basically have different phase relations of the laser throughout the volume of the bead. Whereas in the Rayleigh regime, the particle, say if you had like 100 nanometer bead, which is 1/10 the size of the wavelength of a 1 micrometer laser, then the whole bead acts as if it's in one static uniform electric field. Does that make sense?
Yeah. So what's typically like in lab, what kind of wavelength typically people use? Or
a wide array. It's actually generally in the visible range, mostly, at least in my lab, because we want to work with cells and living things. So you want to stay in the range of wavelengths that won't damage anything that you're looking at.
I see.
But you can do this in any, just about any wavelength you'd like, as long as the forces are strong enough to overcome the weight of the particle or the thermal forces that the particle is experiencing being in some kind of fluid, whatever you're imaging in it. But in my lab, it's generally like a micrometer size bead and lasers from 400 nanometers to 800. We also have a 1064 nanometer. So everything's about a micron.
I see. Okay. Gotcha. Thank you.
No problem. So this is an explanation of this is the geometric optics explanation of what's going on. So this is actually another diagram from Ashkin's paper. And you can see this little curve here is representing the intensity of the incoming laser. And it's usually assumed to be Gaussian. So in a Gaussian, obviously the center has the most intensity and the tails, the intensity decreases as you go away from the beam axis. And then I just want to draw your attention to these 2 points, A and B. So these are kind of thin sections of the laser. A has a higher intensity than B. So we can follow these beams through the sphere and then see how momentum is transferred to the sphere. And then ultimately the net effect, we can see what the net effect will be. So when A enters the sphere, it refracts and changes direction. So it refracts and goes downwards. So the equal and opposite reaction is actually for the bead to react upwards. So you can almost think of it like bouncing off, some molecule holding the bead together right here. And then a similar thing happens at this interface where it refracts and it actually bends down again, and that will cause another reaction in the opposite direction. On the B side, everything's mirrored. So B goes up, the bead is pulled down, and then at this point, the bead goes up and the bead's pulled down again. But since A is greater than B, there will be a greater force towards the beam axis, causing the whole bead to move towards the axis of the beam, which is towards greater intensity. Now, these forces aren't perfectly perpendicular. They're kind of slightly tilted towards the beam direction. So these also do contribute to some of the radiation pressure during this refraction. And then also the reflections of some of the photons bouncing off the sphere will also contribute to radiation pressure.
Can you say again what's the rate? You say this angle tilted a little bit due to this radiation pressure.
Right. So
what is radiation pressure?
Oh, okay. So radiation pressure is just the name given to the fact that photons can exert force on objects because they have momentum. And then pressure is just force divided by area.
I see. Okay.
So if you ever remember like solar sails, that's the mechanism these work off of.
Just change in momentum of the photon and give.
Right.
Okay.
And in terms of the angle, I'm kind of nitpicking about the definitions because we talk about gradient force and radiation pressure. Radiation pressure is usually thought of the thing that pushes the bead along the axis of the laser, and the gradient force is the force that pulls it towards the axis of the laser. But, decoupling them is mostly just conceptual. The gradient force doesn't have to be radial. And what I was trying to say in this picture is that if you kind of just look at how A bends like this, and you can see this force arrow, you can see it's not exactly, radial. So there is a little bit of a push as it bends through, if that makes sense. So most of it contributes to pulling it towards the laser, but some of it also will push the bead. It's a small distinction. And in this case, the index of refraction of the bead is actually larger than the index of the refraction of the surrounding medium. If it's reversed, you actually get the reverse scenario. So this is a little picture I drew. And I didn't indicate whether red was higher intensity than blue. But the main thing is that instead of, for instance, if we follow this red line, instead of it bending towards this normal line here, it bends away. And as it bends away, the photon goes up. That means the bead goes down. So if red happened to be larger intensity than blue, the net effect would actually be to push it away from the laser. So that's one way where you wouldn't get trapping necessarily. Okay. And this is kind of just a summary of that. In both cases, the particle is pushed towards or away from the beam axis through refraction. That's the gradient force. And in addition, the particle is pushed in both cases by reflection and a component of refraction radiation pressure. We all good?
Yeah.
Excuse me. So here are the equations for the Rayleigh regime. So this comes about from treating the particle as a point dipole. And that makes sense because in the Rayleigh regime, the wavelength is much larger than the size of the particle. So you can approximate the particle as being a point dipole. And through its polarization, you get these two forces. So you have the gradient force, and it's proportional to the volume of the particle. This combination of m's comes from the Clausius-Massotti relationship, and this has to do with the polarizability of the particle. And then you get this gradient of the intensity. So this is the part mathematically where you can see that the force will be towards highest intensity. But let's look at m real quick. So m is actually the ratio of indexes refraction of the bead and the medium. So in the case where the bead's index is larger than the medium index, this whole constant is positive. So the gradient of the intensity will have the force pointing in the direction of the beam axis. If m is less than 1, this term becomes negative and it'll actually push away. And then you have the scattering force. So this is the scattering force in the Rayleigh regime. If you had the Mie regime, you'd have extra scattering coefficients. But in this case, you can see the k to the 4th dependence that we're used to when we learn about why the sky is blue. And then you have this relationship that was here, but squared. And then it's proportional to the intensity. So higher intensity, more radiation pressure. And then in this case, it actually doesn't matter whether the bead has a higher index than the medium, since this portion is squared. So no matter what, the bead's always going to get pushed by the laser. But depending on the relative index, you could have an attraction or you could have it repelling.
So K is the wave number, right? Just proportional
yes.
to frequency. Okay, good.
All right. So I also want to mention, light doesn't only carry momentum, but it can carry angular momentum. So you can use these same techniques to actually spin things. So you can set up a laser to have some angular momentum either through a beam called a Gaussian Laguerre beam. You can generate beams that have elliptical polarization. So it has a rotating polarization, and you can see how that would rotate something. And more generally, you can create vortex beams. All of these have angular momentum and will exert torque on any body that the laser hits.
Can I ask a question?
Yes.
Why photon has momentum? Why?
Why does photon have momentum?
Yeah, photon's supposed to.
Well, so in classical physics, as you know, P equals mv. So you would expect a particle with zero mass to have no momentum.
Yep.
But special relativity unifies energy and momentum. So a photon's momentum actually comes from its energy.
Yes.
Another way of looking at it is if you just you can look at it classically. You know, electromagnetic fields have momentum in themselves, and this can be gotten from the Poynting vector, doing E cross H, and that gives that's just one other avenue of understanding how it has momentum.
Oh yeah. Oh yes, I totally forgot that. Yes.
Yeah.
Okay. I just have. So photon spin is 0, right? So some I kind of contradict myself.
Let me try to remember myself.
The spin is 0, but it can have angular momentum. Why?
Yeah, it can have orbital angular momentum.
Oh, okay. Yeah. Okay.
Which I don't fully understand myself, but it does. It does exist.
Yes. Exactly. Thank you.
No problem. And in addition to that, if you have like an extended object, like some long bacterium, and you trap the head of it, even if your trap doesn't have angular momentum, just based on the asymmetric trapping of this extended object, it could end up rotating, kind of like blowing on a fan, if that makes any sense. So that is basically that for torque. I'm not really going to talk about torque anymore. I guess I am. So this is a way of generating it, which might go over your head, but there's basically this tool called a spatial light modulator, and it's basically an LCD screen, which you can use to edit the phase of the wavefront of the laser on an array. So you have, the tiny little liquid crystals, and you can send voltages through whatever program you decide to use to edit the phase of the plane wave that you send down it, or Gaussian wave, or what in this case, in realistic cases, it would be a Gaussian profile. You edit the phase front, and then you can turn it into any kind of shape that you want. And in this case, they have the phase here in B as a function of X and Y. And you can see how it kind of gives it this, rotational effect, where I guess white would be like highest intensity and black would be lowest intensity. Not intensity, helicity. You can see the bar right here. So 0 phase is black and 2 pi phase is white, and you can see how it oscillates around in the circle. And that creates these little vortex beams here, as you can see in C. And in D, they have a still image of a particle rotating. So you'll have to take their word for it, but it does happen. So that's just so you know that you can exert optical torques. So the main uses for optical tweezers are for measuring forces exerted by single molecules, and this generally is just proteins, and there's billions and billions of proteins, unique proteins that have unique properties that you can probe using optical tweezers by attaching some portion of the protein to a bead. You have the bead trapped in a laser, and then you can attach either a bead, or you can fix the protein to a surface, any way to kind of fix two ends on the protein, and then you can start pulling on it, and you can figure out the forces exerted by the protein in that configuration. Or you can do things like measuring codon lengths and transcription dynamics of RNA and DNA, and RNA polymerase and DNA polymerase, and all sorts of protein machines. And more generally, just micromanipulation in live cell environments. And at the moment, these are generally done with transparent beads, roughly a micrometer in diameter. And Now, you can't just take like a glass bead and then attach it to a protein. You need some kind of molecular linker to attach the two, but that's kind of biochemistry, and I've never done that personally, so I don't know exactly how you would do that, but there are tags, or there are linkers you can use to attach a protein to a glass surface, for instance. Okay. So here are some methods of how to calibrate the force. So everything's a spring, so you assume that the bead kind of the bead being trapped by the laser can be thought of being as in a potential well, and you just kind of assume that it's within the linear regime. Now, even if it's not, it doesn't matter because you're getting k as a function of x. So it doesn't necessarily have to be linear, but this is the first assumption to use. And then there's a couple methods you can do. One is to use the drag force of the medium. So you can have a bead in water. You know from the manufacturer the size of the bead, and you know the viscosity of water. And then you can use a piezo stage to move the sample and the water in it, and the bead will be held fixed, and you're basically dragging the water across the bead, and that will pull the bead slightly out of the trap. So the balance of the hook force and the Stokes force will give you an equation for K. You can measure X basically just by looking through the microscope. There are various methods of doing it, but you can just measure X as long as you know the distance scales. And then V is something that you control, and ultimately you can get K from that. So that's a very basic way of measuring the spring constant of the laser. Well, it's really spring constant of the laser bead system, because if you have a different kind of bead, you're going to get a different kind of spring constant. Another way of calibrating is to use the power spectrum method. And what you can do is just keep the bead in the trap and then monitor the variance in position over time. And you can take the Fourier transform of that and you fit it to this equation, which this comes from actually Newton's second law. So it's Newton's second law. In this case, I believe, yeah, Newton's second law with the spring force, a drag term, and then there's a random Brownian term. And if you take the Fourier transform of that equation, you get this equation. And if you take the Fourier transform of your data and fit it to this equation, you can extract this parameter called the corner frequency. And the corner frequency is a combination of the spring constant and gamma, which is the viscosity. So you can extract K from this method. And this is usually the preferred method. It's more accurate. Okay. So here's an example of an experiment done using a trap. So we have these two traps here. And let's see. So, looking at this at the moment, if you look at this pink portion, I don't believe that is all that is there. But anyways, they attached the bead to RNA, and they have another, I believe, RNA handle here, which is attached to RNA polymerase. And RNA polymerase is what basically copies RNA to create more. And as the RNA pulls, or as the RNA polymerase pulls in the RNA, it'll exert a force on this bead, which you can then measure. And it has some interesting qualities to it. So you don't have to read this description, but this is just the result of this data. I would say the most noteworthy thing is this graph here, B, which I increased in size. And you can see there's these little plateaus that occur, these little dwelling regions as the RNA polymerase pulls through the RNA. And with optical tweezers, you can actually measure this activity. So I think that's pretty cool. And you can actually characterize, based on what you know about the molecule beforehand, what's occurring at each of these stages. This number here, 239 BP is in base pairs. So you can actually measure this effect at the resolution of the individual base pairs of RNA.
Can you say again BP?
BP stands for base pairs. So like, if you imagine a DNA molecule, it has that base pair sequence, and yeah, so you can actually measure this at that resolution.
So you mean as a function of time, this RNAP will try to increase the RNA, is that? And they'll exert the force.
Yeah, basically. So RNA polymerase's job is to copy RNA. So the polymerase part is like, it's polymerizes it. It's making a new RNA by polymerizing components in the surrounding medium. And you can monitor how it's doing that step by step by seeing how the beads are being pulled during this process.
So why during this process the force will increase?
Well, so if we go back to the diagram,
yeah.
So this kind of darker blue is the RNA.
Okay.
Or actually, I think this is DNA. My bad. I'm not a biologist. So this is DNA, and I think the RNA polymerase is making RNA from this DNA. Regardless, what's happening is, so this bead is trapped with this laser, and then DNA is attached to the bead, and it's being fed through this RNA polymerase. So as the RNA polymerase starts pulling the DNA through to, copy different codons, different base pairs, it's going to exert a force as it pulls it through. So it, as it pulls through, it'll stop, and then you'll know that the force is, stopped increasing after a certain amount of time. Then it'll keep pulling it through so it can keep copying, and that's basically why you get the signal.
Oh, I see. Okay. Good.
Yeah.
Wow. Interesting setup.
Yeah.
So how they come up with this?
It's pretty crazy that they can actually do this.
How do you come up with this kind of idea? I don't know. Okay. Wow. Okay.
People are amazing. Yeah, it's basically, if you imagine, this might be a bad analogy. If you imagine RNA polymerase is like those tape-fed computers back in the day. They used to feed tapes to do calculations. So that's RNA polymerase is that computer. The tape is the DNA, and then the bead is you holding on to the tape to just feel how it goes through, so you can try to decipher anything about it.
Yeah.
So here's another example, which I think is...
I have a quick question.
Sure.
So if you go back to that plot, what are the like 5 different different color plots or different color lines on that?
So let's see. So there's this assisting force here. I have to remember correctly because there are these 2 beads here. I'm pretty sure the assisting force is the force exerted by this top bead here. So this bead is kind of just measuring the force of the RNA polymerase kind of pulling through. And then this is an extra force to kind of stabilize everything, if that makes sense. Because if this wasn't here, this would be kind of like floating around randomly.
Oh, I see. Okay. That makes
Yeah,
sense.
I have to admit, I don't know the full details of why these different assisting forces produce these different curves. Um, it kind of seems just by looking at it that less force gives you more variance. Like you can see that this line is a lot more noisy at 7 piconewtons versus 25. But I'm not sure how that affects the dynamics of RNA polymerase.
Got it. That makes sense. So are those plateaus just where the process kind of takes a break for a few seconds and
Yeah, and the interesting thing about those parts is that usually has to do with some kind of conformal change of the RNA polymerase, because like this picture doesn't do it justice. These things are these crazy like folding machines that, you know, I don't know much about them. Once again, not a biologist, but you know, depending on its step in the process, it might need to like unfold and rearrange to get to a position where it can continue, you know, copying DNA into RNA. So those little spots are indicative of conformal changes of the RNA polymerase folding and twisting and whatnot.
oh, okay. Cool.
Yeah. All right. So I think this is a much easier example. Maybe I should have started with this. But this is a schematic of a bead attached to myosin. So myosin is a protein in your muscles, and it's crazy. These things literally look like legs and feet, like actually. And then this fiber, that this double helix fiber, is actually actin. So these are these long fibers in your muscles um, that are basically the structure of your muscle. Well, that's they're a major part of the structure of your muscles. Muscles are very complicated, but these myosin actually pull other parts of the muscle along this actin fiber for contraction. And you can actually attach a bead to these myosin, and you can actually look at how it moves along. So you can kind of get some hints of what might happen just by looking at the structure. So you have these two legs and you have these 2 feet. So first the feet has to attach to the actin, then it has to lift up, pull over, and then reattach, and then repeat that process. So if you look at the data here, A is kind of the overall result, and then B, C, D, E are or B, C, D are kind of zoomed in portions. So this is position versus time. And you can see it kind of has the same stepping motion. And if you think back to what I just said, that it has to like detach, then kind of move over, and then it can, and during that moving over, it pulls and then it attaches. And then, you know, it has to repeat that cycle. You can see that during the times where it's attaching and detaching, you have these lulls, and then you have these big jumps when it actually moves along again. And I believe the reason why it stops abruptly here is because you're actually pulling on the myosin in this case. So it's not really in its natural environment. Like you have it attached to a bead, and it's probably moved to a position where, you know, it tried to pull the bead along with it, but it couldn't resist it anymore, and it kind of got slung back, and then it restarted. So you're torturing the myosin here. It does these little steps, and then once it gets too far, it pulls back. So that's what's happening with these big gaps. And then it just kind of wobbles along in these little steps. So you have billions of these in your muscles, pulling at your muscles every time you contract your fingers. And then, yeah, you can see the kind of zoomed in B and C, what the little hop looks like. And then I believe D and E are also examples of this. And then F is the perpendicular position. I'm not exactly sure why they showed that, but you can see it's not really hopping that much in the z direction. I guess that's good to know. So that's another use of optical tweezers for measuring force. You know, it's very narrow use case, but it's limitless because, you know, there are so many different proteins in the bodies that do different functions that you can measure using this method. So the next step.
Sorry, I got one more clarifying
Sure.
question. I'm just kind of, I'm interested. So these myosin, they move, if you go to the last slide, they move like, they move, you know, hundreds of nanometers over the course of like a second or so.
Yeah.
How the heck does that, like, does that very small movement, how does that net result in like me being able to like flex my arm in, you know, a fraction of a second?
So I think, yeah, that is a great question. One thing to consider is it's not in your muscle, it's not just one myosin molecule doing this. It's billions all at once. And when I say all at once, it's not like in lockstep where they're marching perfectly together. This is a random process that occurs in the presence of specific molecules like ATP that cause this motion to start.
Okay, yeah, I guess that makes sense. I don't know, I just think it's pretty cool, pretty amazing.
Yeah, that is, I never thought about that, to be honest. But yeah, I mean, if you have billions of these moving, you know, towards the same direction, but at slightly different times, I can see how after a second, you know, a billion times a nanometer, like you could move 100 meters with your muscles. So it's very plausible that this could occur. And yeah, the way that it started, I'm sure there's, you know, some neuron activity that causes the release of some kind of chemical. I don't know anything about the biology, but yeah, the collective motion.
So the position here is first you calibrated the spring constant and all the force, and then you transform the force into this position of displacement.
Well, in this case, you can actually just measure the position directly.
Oh.
So actually, when you calibrate the optical tweezers, actually, before I explain this, I should mention they do measure the force, but none of the graphs here are of force. And when you calibrate the tweezers, it requires so that gives you force as a function of position.
Yeah.
So when you do your measurements, you require position and you require displacement of the bead from the beam from the laser's center to then translate it into force anyway. So you're always measuring position first.
Oh, I see. Okay. Yeah.
Or displacement rather.
Yeah, makes sense.
In this case, so if you remember at the beginning, I talked about like fluorescently labeling particles. That could have been done here, actually. You could have fluorescently labeled the myosin, and then you could actually see literally in real time it moving along the actin fibers.
How to label?
So I don't know how to like do it from start to beginning, but there are recipes. But basically, generally you need to alter the biology of the protein, which is kind of a problem. So you need to make it available, or you need to make it so it can accept attachment to a fluorescent molecule, which you would put in the sample. So you have to genetically engineer the myosin to either express this fluorescent molecule or accept it as an attachment, and then you basically have this mutant myosin that hopefully works pretty closely to what you to the normal version. And then yeah, okay, so it's almost 7:30. I did not expect it to be, I expected this to be short. I'm like halfway through, so I'll just keep going. So the next step in this for us, because, you know, it's actually a good segue talking about fluorescent labels. I personally am not comfortable with that. And also, if you want to image a cell, you know, you can't label every single protein. You know, if you want to, you know, image this cell in real time, you can't, you know, genetically engineer the whole cell, and there might be unintended consequences when you do that. We always hope that through that process it doesn't affect the function, but you never know. So the tool that has been developed recently is to use metal nanoparticles as chemical probes using Raman spectroscopy. So the plasmon resonances on the metal nanoparticles allow for the electric field enhancement of the laser that you're probing with. And the Raman signal itself is chemical specific because it depends on the vibrational and rotational modes of the molecule that you're interested in. So, and generally in this field, people are looking towards gold because it is the most stable and it has plasmon resonances within the visible region, which, you know, cells like to live in. And some people are using, like, there are multiple different materials that are used, but I think gold is the best. You know, some people are using quantum dots. Some people are using aluminum for the UV portion. Silver is also used, although I think silver might have a problem with cell toxicity because it's actually an antibacterial. People actually use it in the medical field to, like, sterilize wounds. So it's not ideal, but gold is good. So the hope is to be able to use these probes to image the whole cell. So what is Raman scattering? So it is inelastic scattering of light off of molecules, which basically means energy is not conserved. Now I have this diagram about virtual states. I don't fully understand the quantum description of Raman scattering, but there's a much easier to grasp classical description that has to do with the vibrational modes. So you can imagine if you have just any atom really, and it's stretching, or how about carbon dioxide? So carbon dioxide is a linear molecule. So you have carbon, and then you have two oxygens, and you can imagine it can stretch and contract along the length of the molecule. So as it contracts, you can kind of imagine the energy actually increases. And when it's excited with light, when it's in the contracted position, it will let off light with a higher energy because, you know, all of the energy levels are kind of compressed together. And in the opposite scenario, when it's stretched from light hitting it and it stretches, if it releases a photon in the stretched position, it will release a photon of lesser energy. So there's some math involved in that. If you look at the polarization, first order, it's just proportional to the electric field. Alpha is the polarizability, which is a material dependent quantity. And then E bar is just the electric field, so just normal plane wave electric field. And you can plug it in. And then you can assume this dq being an infinitesimal displacement. If you look at the diagram, you can see there's L minus Q0, which corresponds to the compressed state of the molecule. And then there's the L plus Q0, which is the stretched. And this is, what's the word? This is shown here by having Q0 cosine 2 pi nu vibrational, which is the natural vibrational frequency of the molecule. And then you could do a Taylor expansion of alpha, first order, And as long as this d alpha dq derivative is not 0, you'll get this effect. So you can plug it in, and then you can do some trig identities. And then the most important thing are these last two cosines. So this will be kind of the oscillation of the polarization, and oscillation of polarization leads to re-emission of light. So one of those is the frequency of light minus the natural vibrational frequency, and the other one is the frequency of light plus the natural vibrational frequency of the molecule. So since the emitted light depends on the vibrational frequency, you'll get chemical specific signals from this effect. Does that make sense? So the problem with this is this signal is usually very weak, 1,000 to 10,000 times weaker than Rayleigh scattering. But you can help that by so-called lightning rod effect. So the technical name for this is Surface Enhanced Raman Scattering. And what you can do is you can create nanoparticles that have sharp tips on them, kind of like lightning rods. And those lightning rods will enhance the electric field up to 10 to the 8 to 10 to the 11 times stronger, which is certainly enough to create a measurable Raman signal. You can also have a particle dimer, and a particle dimer is basically where you have two metal particles next to each other, and within the gap, you'll get field enhancements. So that arrangement is called a dimer. So this effect is enhanced due to the local surface plasmon resonance. So it's really a resonance effect in addition to this lightning rod effect, which creates such a high intensity signal. The plasmon resonance is a function of the nanoparticle size, shape, material, external dielectric properties, so the medium that it's in, and inter-nanoparticle separation in terms of dimers.
Can I ask a question? So
yeah,
why it's kind of counterintuitive? I mean, the surface roughness, if it's rougher, then it seems like they are kind of interference, so there'll be like decrease the field. So
I'm not so sure. Once it hits the conducting surface, I don't think any optical interference effects are prominent, but I don't know for sure. The baby picture for this has basically do with electron density, or with charge density rather. And this little Microsoft Paint picture, I think, illustrates that. So if you have an E field coming in, it'll separate the charges on a sphere kind of like this, evenly distributed. But when you have these sharp points, the charges kind of get pulled into a more condensed region. I don't know if that's extremely satisfying, but that's kind of the idea. And that's also why the term lightning rod is used, because when you have a sharp tip, it tends to be that if you have like a sharp tip attached to the earth, it's an equal potential with the earth. So the charges kind of come up into the tip to equalize the potential, but because the charges are focused in such a sharp point, excuse me. I don't think that makes sense, actually.
Yeah, I'm just wondering, like when light scatter and then they'll kind of destruction interfere with, maybe. I mean I don't know.
Well,
if
we can.
I don't know.
One thing to consider is that these particles are much smaller than the wavelength of light generally.
Oh.
So there might be those issues if maybe we had like an X-ray hitting
Oh,
you know, this gap, and then like you say, there's some kind of interference effect, and because the frequency is so high, like you start to get these optical effects. But if we imagine the Rayleigh regime, so I have like what 1 micrometer wavelength, and this is like a 10 nanometer particle, you can basically imagine it just like a static electric field pulling these charges apart. Is that good?
Yeah. And what's the plasma? I mean, how? How do?
So plasmon is a name for a quasi-particle, which means, you know, it's not like a fundamental particle, but it's a collective excitation of electrons. And that collective excitation of electrons has, you know, particle-like properties. It has its own energy. It acts like a particle. Well, it acts like a wave in a particle. You know, it has all those quantum properties that makes it seem like it's its own particle. So that's why it's called a plasmon. But it's really just an electron wave. And actually, the waves. So there's plasmons on the surface and there's plasmons in the bulk. So the electron waves that occur on the surface are actually longitudinal waves. I wish I had a diagram for it, but you know, you'll have plus, then minus, then plus. Like if this was like a surface right here, you'd have plus, then minus, then plus, then minus, oscillating along the surface.
Yeah.
And in the bulk, I'm pretty sure there are a combination of transverse and longitudinal, but I actually am not sure of that. I only know about the surface plasmons.
So surface plasmon. So I guess it's transverse. So their frequency will be different. Is that transverse and longitudinal?
There's actually a neat little derivation you can do involving the polarization of a material.
Yeah. And
you can derive what's called the plasma frequency from that, which is basically the resonant behavior of the material.
So so basically this frequency for gold is just the frequency is just y, the visible y around that frequency.
Correct.
So that's why we prefer to use gold. Just we'll see a huge signal.
Yeah, you'll see a huge signal. And what's cool about these, I wish I had a picture in here, is you can actually see it change color, like in the microscope, pretty obviously.
Okay, good. Understand.
And here's some diagrams for gold. So AU is gold, and it's a 100 nanometer gold sphere. So the left chart is the polarizability, the real part of the polarizability. And it shows that around 600 nanometers of wavelength that there's this peak here. So this corresponds to the plasmon resonance here. And B, these are the scattering and absorption coefficients. And you can see at the same 600 nanometer, the scattering peak is maximized, and then the absorption peak, it's a little bit blue shifted to that peak, but you can see how these align. So you might want the configuration I imagine is that you would trap the gold sphere, say at around 1,000 nanometers, so you don't have huge absorption and scattering. And then you could have a probing laser, a second laser, that would excite this plasmon resonance, so you can activate the sensing property of the molecule or of the nanoparticle. Does that make sense?
Yep.
Okay. So here's an example of this being used. So these people, they're trying to make a new glucose sensor for diabetics. So they tested this sensor in vitro and in vivo in rats. And basically they use silver-coated polystyrene beads on a titanium substrate. So in this case, they're using the dimer amplification. So you have a bunch of beads in an array coated with silver, and since they're close to each other, you'll get this amplification effect. They use these two molecules, 1-decanethyl and 6-mercapto-1-hexanol, to attract the glucose. I don't know how that works, but that's how they got the glucose to come close to the sensor. Unfortunately, this creation was only stable for about 10 days in bovine plasma. And once again, their goal is towards a continuous glucose sensor for diabetics, because right now they just have to like prick their finger periodically to get their blood throughout the day. They're trying to work to make something that you could just wear and then it will continuously monitor the glucose in your blood. So this is how they built it. Basically, as I described, they have an array of these spheres coated in silver, and then they use these molecules to pull in the glucose. So unfortunately, I don't think you'll ever see this used in a human. They basically did surgery on this rat and then put this device in the rat and then attached the rat to this whole laser setup. And they used a Raman spectrometer to get the Raman signal out. And this is the signal you get. So this square wave at the top here, you can see it's divided into 4 sections, A, B, C, and D. Oops. And that has to do with the glucose concentration. So at A and C, there was 0 glucose. And at B and D, they had 100 millimolar of glucose. So when you pump that in, you get these 4 signals. And then you can subtract combinations of those to see if you can detect the glucose. So these are all Raman signals, and you can see they're measured in wave number shift as opposed to frequency. You know, they're related. I don't know why they use wave number shift, but wave number is proportional to momentum, so maybe that's why, but I don't know. But you can see for F, for instance, they subtracted A from B, so that should show the signal where there is glucose present. And then D from C is a similar scenario. So D has glucose and then C has no glucose. So they're subtracting the background, and you can see these sharp peaks. And they pretty much match the reference glucose. And then they, just to check, they did C minus A, which both C and A are background, and you don't get much of a signal. And then D and B, if you subtract those, you get nothing as well, because those are the signals where you have glucose. So this is just SIRS in action. And then they compared it to the normal blood prick method, and the results followed pretty well. So luckily this is the end because it's already 7:41. So our goal is to basically combine these 2 methods. So here's a cell. This star is a gold nanostar. These are 2 red lines, are a trapping laser at, infrared wavelengths. And then these green parts are meant to excite the Raman signal. And the hope is I could take this star and raster scan it, point by point at whatever I want to look at really fast. And I should be able to get real-time Raman signals in a live cell. In theory. There's a lot more technical aspects to this, but that's basically it. And that's the end of my presentation for you.
So every time we can, you mean, so we just have many nanoparticles on, say, the whole cell.
Mm-hmm.
And okay, so we can image every part of, in principle, we can image every part of the cell and see how just it moves or like things like that.
Yeah, at the chemical level, hopefully. So we'll be able to sense when chemical reactions are taking place as well at different parts. And it's actually pretty easy to achieve multiple traps using one laser. So it's not a very difficult endeavor. You basically, if you remember that SLM that changes the phase, you can change the phase such that it gives you as many traps as you want.
I see. So okay, I forgot my question. Sorry, I forgot my question. Okay.
No, it's okay. Take your time.
Okay, so this is your last slide.
Yeah.
Okay, thank you. So see if Chad and Reina have any question.
Yeah, did I bore you guys?
No, I think it's interesting. I mean, this is my first time to really see or hear a talk about biophysics, actually.
Yeah.
I don't know.
What were you say? I'm sorry.
No, I mean, I never heard a talk and never thought of kind of how to combine all this together.
Yeah, it's really nice. It's basically putting a whole bunch of well-established things together and getting something new out of it. Just nice. Whereas I was going to say what differentiates me from like a theoretical biophysicist, they're like going crazy with like computational simulations of like protein dynamics to like I'm being hyperbolic here, but like literally figure out how a protein works from QED or something like that. You know what I mean? So this is kind of like the application side of biophysics.
So what's the like? Oh, okay, I remember my question. So the chemical reaction, what's the typical time scale for that?
Ooh.
So my real question is that, so I mean, can we resolve those reaction time? I mean, compared to our, I think it's kind of depends on our light, our incoming laser, and then it'll tell us if we can resolve this small time, the time resolution of this technique. I think that's my question.
So I feel like your question's asking like if you can see like a chemical reaction happening in real time, like like instantaneously. When I say real time, I don't mean instantaneously. I mean you can kind of see like the before after, because like although you may be interested in a single chemical reaction, I would say the first step, if you want to just do this generally in a cell, is you can kind of just see you know concentrations building up in this chemical reaction. But to actually answer your question, I'm not sure about the timescales. I don't think there's a theoretical limit that would make it impossible, but I think the temporal resolution has to do mostly with the equipment in terms of how fast your camera can actually sense you know the signal versus how fast the chemical reaction's occurring. I actually don't know the answer to that question, how fast a chemical reaction takes. That's actually an interesting question. I'm going to write that down.
No, just wonder. Okay, thank you so much. Yeah, so
no problem.
So if Chad and Rena, you have question, you can raise.
Probably go.
Okay, if not, then yeah, then thank you, Phil. Then yeah,
no problem. Thank you very much for having me.
Good luck with your teaching, research, and qualify. Okay.
Thanks.
Hopefully next time we see each other, we
both pass.
Both pass and about to become just the candidate.
Yeah.
60%, 80% candidate. Just finish the courses like that.
Yeah. Okay. All right.
Yeah. Okay. Right now. Okay. So I'll see you guys later.
See you guys.
Bye bye. Good night.
Good night.